Good afternoon, students. This is Reema, your biology educator from V-Learning. As you know that V-Learning is an online educational platform where we strive to provide quality education for all. So to get all the benefits from our channel, if you have not subscribed to our channel, do subscribe to it and also share it with your friends so that they will get benefit out of it. And don't forget to visit to our website, that is www.v-learning.in, where you will be getting all the live classes, pre-recorded videos, animated videos, and the PDF notes of all these chapters. So without any delay, let's begin today's session. So what we have learned in the previous class? In the previous class, we have learned about complex permanent tissue. Under this, you have seen that there are two types of complex permanent tissue. One is your xylem and phloem, right? And where you have seen that xylem is made up of again 4 different elements, right? That are your tracheids, vessels, xylem parenchyma, and xylem fiber. And also you have learned about that the phloem, it is also made up of again different types of elements, and they are responsible for performing different functions in case of the plants. And what are they? They are also what? They are your vascular bundle, right? So, and also we have learned about the tissue system. Did you remember that we have learned about the epidermal tissue system, where you have seen that the epidermis, which is the outermost part of the plant, that is, it is the epidermis, and there we find the stomata, and in the stomata, what is present? That is your Gerd cells are present, right? And this Gerd cells are responsible for your opening and the closing of the stomata. And you have seen that the inner wall of the Gerd cells are thicker, right? And the outer wall of the Gerd cells in stomata are thinner. So, and we have covered about your ground tissue system. What is this ground tissue system? This is nothing but ground tissue are all the tissues except your epidermal tissue and the vascular tissues. So all the tissues which are coming in between, we call them as ground tissue system. Like your parenchyma, sclerenchyma, collenchyma, all these are your ground tissues. Alright. So what we are going to learn in today's class, in today's session, we are going to learn about the anatomy of dicotyledonous and the monocotyledonous root and shoot. And also we will be looking into the vascular tissue system, which was the last tissue system, right? So we will be covering the vascular tissue system and also the anatomy of the dicotyledonous root and shoot. As you know that the anatomy is nothing but the internal structure, right? Internal study of the root. So we will be looking into what is present inside the root. If you see inside the root or the stem, the TS, that is the transverse section of the root and the stem, how it will look like, we'll look into it, okay? So let's begin today's class. This is your lecture number 3. Yes, so first of all, we'll be learning about the vascular tissue system. So as you know that vascular system consisting of 2 things, that is your xylem and phloem. So xylem and the phloem, which is your complex permanent tissues, they are constituting your vascular tissue system. So as you can see here, the xylem constitutes of the complex tissues, that is the phloem, the vascular system constitutes of the complex tissues, that is your xylem and the phloem, right? So these two, that is your xylem and the phloem, it is constituting your vascular tissue system. So next, the xylem and phloem together constitute the vascular tissue system. So how this xylem and phloem are arranged inside the roots, inside the stem, how they are arranged? So there are again various types of arrangement, all right? So let us see the various types of your vascular tissue system also. So first of all, see, in case of your dicot system or dicot stems, the cambium is present. Now in the last class, I have said that what is cambium, right? Cambiums are nothing but these are your living tissues. So these cambiums are what? These are your living tissues. So where they are present, this cambium are present in between the xylem and the phloem. And what they are doing, this cambium are responsible for the secondary growth in the plants. All right. So this cambium is also responsible for the secondary growth in the plants. So they are also responsible for the secondary growth and the plants. So in case of this dicot stem, only in case of dicot stem, the secondary growth takes place, not in case of your monocot stem. So as you can see, is present in between the xylem and the phloem, and they are the living tissues which are responsible for what? For the secondary growth in the plants. So such vascular bundles, because of the presence of cambium, possess the ability to form the secondary xylem, right? Secondary xylem and phloem. So these tissues, and hence they are called the open vascular bundles. So these vascular bundles again can be categorized into 3 types. Okay. So based on the conjoined vascular bundle, this conjoined vascular bundle is divided again into 2 types. One is the open vascular bundle, another is your closed vascular bundle. So there are basically 3 types of vascular bundle. One is your radial vascular bundle, second one is your conjoined, and third one is your concentric vascular bundle. So this conjoined vascular bundle, this conjoined vascular bundle is again divided into 2 types. One is your open vascular bundle, another is your closed vascular bundle. So in case of this open vascular bundle, cambium is present. Okay. So in case of this open vascular bundle, cambium, it is present, whereas here in case of closed, it is absent. All right. So in case of your that is, in case of your open vascular bundle, cambium is present, which is responsible for your secondary growth. This open vascular bundle only can do the secondary growth, whereas this closed vascular bundle, it cannot do the secondary growth. So as you can see here, right, the phloem and the xylem, they are present in the same radii, right? So they are not present in the opposite radii. So they are close to one another, right? And in between here you can see cambium, isn't it? So this type of vascular bundle where cambium is present, we call it as open vascular bundle. So here you can see cambium is present, right? So which type of vascular bundle it is? In dicot stem, you will see always the open vascular bundle. So here the vascular bundle is your open, and it will lead to your secondary growth in plants. So it will lead to your secondary growth. Whereas the closed vascular bundle, it will not lead to your secondary growth. Whereas in case of your monocotyledonous stem, you will not see the secondary growth, because the vascular bundle is closed and here there is absence of cambium. You cannot see cambium here, right? So see, in the monocotyledonous, the vascular bundles have no cambium present in them, hence they do not form the secondary tissues. So here in case of your monocot stem, they are not responsible for secondary growth. Only the dicot stem can do the secondary growth because they do not have cambium. So cambium is absent here, right? So you see, they are lying in the same radii, right? This is your xylem. Xylem is present inside, all right, and the phloem is present here outside. So xylem and phloem are present in that manner. Xylem is present inside and the phloem is present outside. Next, what are the various types of vascular bundle? The vascular bundles we can categorize into 3 main types. The radial vascular bundle, conjoined vascular bundle, and the concentric vascular bundle. Let us see what are these. So, the types of vascular bundle based on the arrangement of your xylem and phloem. So, if we see the various types, they are of three types. What are the three types? One is your radial, right? Next one is your conjoined, and the third one is your concentric, right? Concentric vascular bundle. So this radial, so how this xylem and phloem are arranged? They are arranged in the opposite direction, okay, towards the radial. So this xylem and phloem here, the xylem and phloem are arranged in opposite direction. They are not together. They are not close to one another. So, if this is a cambium, all right. So, see here, this is your xylem. This one is your xylem. This one is the xylem. So, here, the xylem and phloem are in 4 bundles, right? So see, it is called as tetrach, right? So where xylem and phloem, they are arranged in the form of 4 bundles, they are known as tetrach. So this one is your xylem. So they are in the form of radius, right? So see, this is the xylem and this round one is your phloem. So they are arranged in an opposite direction. So this is your radial vascular bundle. Now, this is your xylem and this one is your phloem. Now, what about this conjoined vascular bundle? This conjoined vascular bundle means the xylem and phloem, they are lying together. They are close to one another. So, here the xylem and phloem, they are close to one another. This conjoint, again, it's divided into collateral and bicollateral. So this collateral conjoint vascular bundle is divided into 2 types, that is your open vascular bundle and closed vascular bundle. Here in the conjoint vascular bundle, it is divided into 2 types again, that is your open vascular bundle and closed vascular bundle. So open vascular bundle and closed vascular bundle. So in the open vascular bundle, as we have seen that here the cambium is present, right? Xylem is present inside and this is phloem, and here the cambium will be present, and this cambium, it will lead to the secondary growth in the plants. That will, it means it will increase the width of the plant or the grit of the plant. So this is your cambium. So here cambium is present. Here in the closed vascular bundle, you can see the xylem and the phloem. Here cambium is your absent. So here cambium we will not see, right? Cambium will be absent and here cambium will be your present. So this concentric means, that means here the vascular bundles are lying one, that is they are lying one above the other. So here the vascular bundle, that is the xylem and phloem, they are lying one above the other. So if this is a phloem, inside this will be your xylem. All right. So if this is a phloem, this will be your this is a xylem, this will be on top of it will be your phloem. So again, this concentric vascular bundle is divided into 2 types, that is your amphibrical and amphibrical and amphibricular. Okay. So again, it is divided into 2 types. So these are the various types of your vascular bundle. So next let us see what is the definition of your radial vascular bundle. So when the xylem and phloem are lying where within, okay, within a vascular bundle are arranged in an alternate manner along a different radii. So they are arranged in an alternate manner, that is in the opposite direction, right? And in the different radii, as you can see, this is your phloem, this is the xylem, right? So that type of arrangement of your vascular bundle is known as your radial vascular bundle. So see here, they are arranged in an alternate manner. Alternate manner. The different radii, right? So this is your radial vascular bundle. So conjoined vascular bundle, as I have said, that it is again of two types. One is your open conjoined, another is your closed conjoined. So in open, you have seen that cambium is present, and here cambium is present, and here cambium is absent, right? In closed. So see, conjoined type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundle. So how they are situated? They are close to one another, right? So if this is the phloem, right? So this is xylem and this is phloem. They are located close to one another. So this is the arrangement, that is in the same radius. So they will be located in the same radius, not in the alternate radius. So such vascular bundles are common in case of stems and leaves. All right. So this type of, that is radial type of vascular arrangement, you will see basically in case of your roots. Okay. And you will see this in case of your stems and leaves. So the conjoined vascular bundles usually have the phloem located only on the outer side of the xylem. So in case of your conjoined vascular bundle, the phloem, that is if this is a vascular bundle, the phloem will be here and xylem will be present inside. Okay, whereas the phloem will be outside, right? The phloem will be outside of the xylem. So this is all about your conjoined vascular bundle. So as you can see, this is clearly given, right? Open and closed. There are two types. Cambium is present here. You can see cambium is present in open conjoined vascular bundle, whereas here you can see in the diagram cambium is absent. So it is divided into two types. Clear? Yes. Now next, let us move into the dicot or the dicotyledonous root and study the anatomy of the dicotyledonous root. So far we have learned about the vascular tissue system, how they are arranged. That is arrangement of your vascular bundles inside the roots or the stem. Now let us study the dicot stem, root and the anatomy of them. So what do you mean by this dicot root? We have studied roots in the morphology of flowering plants, right? What is this root? Root is nothing but the underground part of the plant and which is responsible for absorption of your minerals from the soil, right? And we have learned about that there are three types of roots, that is tap root, isn't it? Fibrous root and adventitious roots, right? So as I have seen that there are three types of roots. Where do you find the dicot roots? Dicot roots that is the dicotyledonous root you will see in case of your tap roots mainly, right? So these are the tap roots, isn't it? So how is the tap root? It is formed from the primary roots itself, right? We see in case of your carrot, radish, we will see the dicot roots. So here it is formed itself from the primary root, not from the secondary root, and the secondary roots arise from here, isn't it? So this is present in case of your tap roots. So in case of tap roots, we will see this mainly the dicot roots, right? And it's consisting of a single primary root. So as I have already said, it is consisting of a single primary root, right? From which the secondary roots are arising. See, all these are your secondary roots. So from here, the secondary roots are arising. And tertiary roots develop and grow vertically downwards towards the soil. So from here your secondary roots and the tertiary roots arise. So this is basically your primary root only. Taproot is your primary root from where all the roots are arising, and taproot you will see in case of your dicotyledonous plants only. So see, next let us understand the anatomy of your dicotyledonous root. So how the internal structure of your dicot root. So let me write here anatomy of dicot root. Let us understand the dicot root with the help of this picture, that dicot plants always will be, that is their stem will be, that is huge in size. Okay, so these are your roots. Okay, let's say these are your dicot roots. So in the dicot roots, the pith will be very small. Let's say, okay, let's say this is the pith. Okay, just remember the pith in the form of a star. You can remember it in the form of a star, a small star. Okay, so these are the dicot tree, that is your dicot trees will be huge. Okay, their stems will be dark brown in color. All right. And whereas, so what are these monocots or monocotyledonous plants? This monocotyledonous plants, their stem will be thin. Okay, it will be basically green in color. So all they will be having the thin stem. In case of your monocot, in case of your dicot, their stem will be thick. As you can see, all the thick plants we see and the stems are dark brown in color. These plants will be your dicotyledonous plants. Whereas you see that where here you will see that is those plants are having the thin stem. They are your monocotyledonous plants. They will be your monocotyledonous plants. So here you have to remember this monocotyledonous plant. Just remember here the pith will be large. So just remember the pith will be large in case of your monocot roots. So let's remember it with the help of a sun. Okay, so see here, just draw it a sun, and here let's draw it a star. Okay, that means the pith will be very less in case of your dicotyledonous roots, whereas in case of it, let's remember this is the pith and the pith will be large. Okay, so they have a large pith. Remember this. Now let me draw the diagram of your dicotyledonous root. So what will be present in the outer part? So see, if you see inside the root, anything, that is any part of the plant will be having the epidermis, right? That is the outer part of the plant. So similarly, the roots also has epidermis. So here let me draw the epidermis. These are all the epidermis. So these are the epidermis. So here the epidermis is called as epiblema. So what we call it as epiblema. Alright. So what are these? These are your root hairs. Okay. So these are the root hairs. So these are your root hairs. Now here what will present after this, here the parenchymatous cells are present and they have got intercellular space between them. So this parenchymatous cells with the intercellular space, we call it as cortex. So these are basically the cortex. So cortex here, it is made up of what? It is made up of your parenchyma cell, and they have got, as you have studied, right, this parenchyma cells, they have got intercellular space, right? So they have got here, as you can see, These are the space in between them, right? These are your intercellular space. So after this cortex, what is present after the cortex? There will be your endodermis. Endodermis is the inner layer of any part of the plant. So after this, they will be having endodermis. So this is your, that is everything from here, it will be your endodermis. So this is, sorry, yeah, okay. So this is your endodermis. This is your endodermis. So outside this layer is your endodermis. So see this portion, everything inside the endodermis, if this is the endodermis, everything inside the endodermis, we call it as stem. Okay, everything, whatever will be present in the endodermis, we call it as stem. So see here there will be another layer, we call it as, this layer of cell, we call it as barrel cell. Now, what is the function of this barrel cell? This barrel cell, it is rich with this cuticle, or it is rich with the suberin, which is responsible for preventing the water loss from the plant. Okay, so here inside this, there will be a layer. That layer, the first layer of your endodermis, we call it as pericycle. This layer is called as pericycle. All right, so this layer, is called as your pericycle. Now inside this pericycle, what is present? Inside the pericycle, your vascular tissues, that is xylem and phloem, will be present. So here the xylem and phloem, it is arranged in the form of radials, that is radial arrangement we will see. So you will see the xylem and phloem here. Okay, so here this is your xylem. Okay, so this one is your xylem, and this, near the xylem, you will see the phloem. This is your phloem. All right. So here can you see pith? This is your pith. Okay. So this pith will be very, very less. Here the pith is very, very less. Okay. In case of your dicot roots. So in case of your dicot roots, the pith is very, very less. Now near this, they will be having some tissues. Okay. These tissues are known as your connective tissues. These tissues near the xylem and phloem. So as you can see, near the xylem and phloem, there will be some tissues. These tissues are known as your connective tissues. They help in the connection between your xylem and phloem. Now as you can see, anything, that is any part of the plant, will be having an outer layer that is called as epidermis. Here we call it as epiblema. So next to the epiblema, we will find the cortex. Okay, so this cortex are made up of parenchyma cell, and they have got what? They have got intercellular space in between them. Now what is the function of this intercellular space? So through the root here, the water gets transported. So from, that is in between, there is a space, right? So through this space, the water gets transported. So as you can see from the root here, the water will be transported, that is not through the cytoplasm, okay, not through the cytoplasm, but from here. Okay, so from here the water will be transported. All right. So this type of that is transportation of water, we call it as Aproplus Pathway. So you will be studying in the later chapters that there are two types of pathway for the transportation of water in the endodermis. One is your simplest pathway, another is your Aproplus pathway. So this type of transfer of water, where the water transfers through the gaps, not through the cytoplasm, we call it as Aproplus Pathway. So why the water cannot transfer? Because here there are the barrel cells, right? So these barrel cells are reached in a waxy layer, okay? They have got a waxy layer that does not allow the water to pass through this cytoplasm. So this waxy layer in the barrel cells, they are made up of suberin, okay? So because of this suberin is a waxy layer which is present here in the endodermis. So here there is a layer of suberin, so which does not allow the water to pass. This is a waxy layer. So because of suberin, they cannot pass through the cytoplasm. They will pass through this intercellular space. Clear? So this is how the dicot root looks like. Now what is this pericycle? So pericycle is the layer which is present inside the endodermis. You can find the pericycle inside the endodermis, right? So it is present inside your endodermis. It is made up of your parenchyma cell or sclerenchyma cells. So here the pericycle is made up of your parenchyma cells. So these pericycles are also made up of your parenchyma cells. So it is the inner layer of your endodermis, okay? And then what is the cell? Okay, what is the cell? A cell is nothing but everything what is present inside the endodermis, we call it as cell. Clear? Now what is this pith? You can see here the pith is very small, right? That's why I have shown here as a star, right? Remember this as a small star, that is your pith. So in case of dicot root, the pith is very small. So as you can see the pith, right? It is very small. So here, yeah, next what we can see, what is the function of your pith? What is the function of pith? Pith helps in the transportation of water and nutrition inside the plants, okay, inside the roots. So this is all about your this dicot root. Let us see what is written in your textbook. The outermost layer is your epiblema, which you have already studied, right? So the outermost layer, you can see this is the epidermis. We call this epidermis as epiblema. So this is called as epiblema. Many of the cells of epiblema protrude in the form of unicellular root hairs, okay? So many of the cells of this epiblema, you can see they will come out in the form of your root hairs, isn't it? So as you can see the root hairs. So the cortex consists of several layers of thin-walled parenchyma cells with intercellular space. So as I have drawn the diagram where I have shown the cortex, right? So there we have seen that this cortex is made up of which type of cells? It is made up of your parenchyma cells, right? And the parenchyma cells will be having your intercellular space. The innermost layer of the cortex is called the endodermis. So the innermost layer of the cortex, we call it as endodermis. So this is your endodermis. As you can see here in the picture, this one is your endodermis. So near the endodermis, inside the endodermis, what we find? We find your pericycle. That is the first layer. So see the single layer of barrel-shaped cells without any intercellular space. So we will find barrel cell. Then the tangential as well as the radial walls of the endodermal cells have a deposition of water impermeable membrane, or the waxy material will find it inside the endodermis, right? What we call it? That is your suberin, isn't it? That is your suberin. What is this suberin? That is a waxy material which we find it inside the endodermis that prevents the water to pass, right? That's why they follow the Aproplus pathway. So in the form of your casparin strip. So this suberin is present in the form of your casparin strips. Next to the endodermis lies a few layers of thick-walled parenchymal cells referred to as your pericycle. So as you have seen in the diagram, what we have seen next to the endodermis, here we have drawn one layer, right? So we have seen the pericycle, isn't it? So next to endodermis, we will find the pericycle. So pericycle is made up of which type of cell? It is made up of your parenchyma cell. Here the pericycle is made up of your parenchyma cell. So they are responsible for storage of the food. So initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. Okay. So what happens in these cells? In these cells, that is the lateral roots and the other vascular cambium, it develops in these cells. And you have seen the pith, isn't it? How is the pith? The pith is large or it is small. The pith is very, very small and inconspicuous. What do you mean by this inconspicuous? Inconspicuous means which we cannot see properly, okay, which is not properly visible. That is not properly visible. Not properly visible. That is the meaning of your inconspicuous. So next, let us see some more characteristics of this dicot roots. So here you can see the parenchyma cells which lie between the xylem and the phloem is called as your conjunctive tissues. So as I have said that in between the xylem and phloem, so as I have said that this is the xylem, this is the phloem. So if this is the xylem, this is your phloem, these are the phloem, isn't it? So here some of the cells will be lying, okay. These cells or these tissues are known as your conjunctive tissues. They are known as your conjunctive tissues. Clear? As you can see, and you can see that protoxylem and metaxylem arrangement here too. So in case of your roots, you have seen that the protoxylem will be lying outside and metaxylem will be inside. We call this condition as exarch, isn't it? So the condition we call as exarch, isn't it? So in case of your dicot root, the protoxylem will be outside, whereas the metaxylem will be lying inside. We call this condition as exarch. So these are usually 2 to 4 xylem and phloem patches. So here you can see this 2 to 4 xylem patches in case of your what? In case of your dicot. In case of your dicot roots, 2 to 4 patches. And later a cambium ring develops between the xylem and the phloem. So here later what happens? The cambium ring also develops. So because of this cambium ring, the secondary growth takes place. So here later what happens? This cambium ring also develops here. Okay. So this is the phloem, this is the xylem, and later the cambium ring also develops in case of this, so which leads to the secondary growth. So all the tissues on the inner side of the endodermis, such as pericycle, vascular bundle, what we call them, everything inside the endodermis we call it as still. Clear? So everything whatever is present inside the endodermis, right? So which one is the endodermis? This purple color full is your endodermis, right? So what we call it? Everything inside the endodermis we call it as still. Is that clear? Okay. This is all about your dicot root. Next, let us move into your monocot roots. Now, what are these monocot roots? Tap roots. That is tap roots you will find in case of your dicotyledonous plants, right? That is dicot roots. In case of this monocot roots, where we will find, that is what type of root we'll find? That is your fibrous roots and the adventitious roots. So in case of your wheat, okay, you will find this fibrous or adventitious roots consisting of a wide network of thin roots. So how are these monocot roots? These monocot roots are very, very thin roots, isn't it? They will be very, very thin roots, and they are arising from where? They are arising from the tap roots, right? That is your primary roots. So they are very, very thin roots, and they are arranged in the form of a mat, right? They are arranged in the form of a mat, and they are very, very thin roots, and they are basically fibrous roots and the adventitious roots. They originate from the stem. So from where they originate? They originate from the stem. Okay, this is your stem. So from the stem only this starts originate. They are connected to the stem. So Now let us study the anatomy of your monocot roots. How are the monocot roots? In case of monocot roots, I have drawn the sun, isn't it? What the sun resembles? The sun resembles the pith. So how will be the pith? The pith will be large, isn't it? So let us draw the monocot roots. So see if this one, so this one is your, this one is what? This one is your outer layer, that is your epidermis. Epidermis. Okay, this is your root hair. These are the root hair. So if we see here, you will find what? You will find the cortex. Here we will find the cortex layer. Just after the epidermis, the next layer is your cortex. Now here what you will find? Here you will find the endodermis, right? This is your endodermis. Endodermis. Now next, inside the endodermis, what is present? Everything inside the endodermis, what we call it? We call it as stele, right? Here only the arrangement of your vascular bundle is different. That is xylem and phloem, they are in the form of a polyarch. That is more than 6. Okay, anything more than 2 we call it as poly, isn't it? They are, that is xylem and phloem bundles. We will find many xylem and phloem bundles. So here that is the pith will be large also. So here what we will find? You will find here pericycle. Right, so let us draw the pericycle. And this monocot roots, they will not do the secondary growth. Only the dicot root will do the secondary growth. Okay, so here we will find the 6, that is xylem and phloem. They will be arranged in the form of 6. And here the pith, this part is your pith, and the pith will be your large. They will be having large pith. Okay, so see, and this is your phloem. These are all your phloem. And similarly here also you will find the connective tissues in between the xylem and the phloem. So only the difference is your arrangement of the vascular bundle, and the pith is large here. So as you can see, there will be 6. Okay, that is more than 2 or more than 6. More than 6 xylem and phloem bundles, and we call this condition as polyarch. And here in the monocot root, we do not see the secondary growth. So here they do not, that is no secondary growth. And pith is your large. You've got large pith. Let us see what are the characteristics of your that is monocot roots. Anatomy of the monocot root is similar to the dicot root. As I have said that everything is similar, only the arrangement of your vascular bundle is different. So it has epidermis, cortex, endodermis, pericycle, vascular bundles, everything is present inside it, right? So as you can see here, the anatomy of your monocot root, right? So as you can see here, everything is present, only the arrangement inside the endodermis will be different. That is, your vascular bundles will be different, the arrangement. So here also you will find everything. We will find the cortex, which is made up of parenchymatous cells, right? So next, as compared to the dicot root, which have fewer xylem vessels. So dicots, they have got very few xylem vessels, that is 2 to 4 xylem vessels or xylem bundles, sorry. So here you will find 6 or more than 6 bundle or xylem bundles, more than 6 xylem bundles. So they are usually more than 6. We call it as polyarch. Polyarch xylem bundles in the monocot root. So pith is large and well developed. As I have shown you how to remember it, like in the roots, you remember a big sun. Okay, that means the sun is nothing but your pith. That means the pith is large here. So pith is large and monocotyledonous roots do not undergo secondary growth, as I have already explained. So next, let us learn about your this is done. Let us learn about the anatomy of your stem. What is present inside the stem, or how the stem looks like. Okay, if you see the stem under the microscope, if you see the TS section of the stem, how it will be. So first of all, tell me, what is this dicot stem? So dicot stems contains vascular bundles arranged in a ring. Okay, vascular bundles are arranged in a ring around the peak. Okay, so they are arranged around the peak. So the stem contains two cotyledon. As we are talking about dicotyledonous, dicots mean always they will be having two cotyledon. That is cotyledon. Sorry, they will be having two cotyledon, right? So, or embryonic leaf called this dicotyledonous stem or the dicot stem. So they have got how many cotyledon? They have two cotyledon. And whereas in case of monocot stem or monocot plant, we will see only one cotyledon. What is this cotyledon? Cotyledon is nothing but it is an embryonic leaf. Alright. So yes. Yeah. Now let us study the anatomy of your dicotyledonous stem. Or simply dicot. Also we can write dicot stem. So what is this all about? How does it looks? Okay. So in case of this dicot stem, how is the arrangement? That means the xylem and phloem are arranged right inside the endodermis. So if this is a cambium, so this layer here we call it as epidermis, right? So in case of this, we call it as what? We call it as trichromes, okay? So these are, that is, this root hairs here, we will call it as trichrome. This is your epidermis. Now, in case of this dicot stem, okay, in case of this dicot stem, their cortex is thin, okay, and it is made up of 3 layers. So, in case of them, the cortex, it is thin, and because of which it is made up of how many layers? It is made up of 3 layers. So, what are the 3 layers of the cortex? The first layer is your epidermis. Second layer is your hypodermis. And the third layer is your endodermis. Alright. So the cortex is thin, so it is made up of 3 layers. So after the epidermis, what is present? So here they have got a layer that is known as cuticle. This cuticle is responsible for preventing or prevent the water loss. Alright, so they prevent the water loss. So inside, what is present? This layer, it is made up of this epidermis. It is made up of your parenchyma cells. So this epidermis is made up of your parenchyma cell, and this hypodermis, it is made up of your parenchyma cell. So it is made up of your parenchyma cell. So this epidermis, it is made up of parenchyma cell, and the hypodermis, it is made up of your parenchyma cell. So inside it, this layer is what? This layer is second layer, we call it as hypodermis. And again, inside this, there is another layer, what is called as endodermis. The third layer is your endodermis. So this layer is your endodermis. Next. Now here you will see the pericycle, right? So in case of this, in case of your dicot stem, the pericycle is arranged in the form of a semi-lunar moon. Okay. So how it is arranged? Let us see. It is not in the form of a cycle. It is in the form of a semi-lunar moon. So this is how the pericycle is arranged. Okay. So this is your pericycle. So this is your pericycle. It is arranged in the form of a semi-lunar moon. So here what we will find inside the pericycle? Inside the pericycle, we will find the xylem and phloem. They will be arranged in the form, right? So they will be arranged, and also we will find what? We will find the cambium too. Okay. So this is your cambium. We will be finding here the cambium too. Okay, so see how it is present. So this is your phloem. This is your phloem and xylem is present inside. Xylem, and this is your phloem. This is xylem. This is phloem. Phloem is present outside, right? So this here some rays will be present. These rays are called as your medullary rays. So here you will find some rays also in case of your dicot stem. These rays are called as your medullary rays. These rays are called as medullary rays. So it is arranged in the form of a semi-lunar moon. So this pericycle, as you can see, this pericycle is made up of scale chimatous cells, not parenchymatous cells. So this pericycle is made up of scale chyma cells. Now why it is made up of scale chyma cells, not parenchyma? You know, the scale chyma cells are having dead cells, right? So the stem, it needs support, it needs protection. If it is made up of parenchyma cell cannot provide support, right? So it is made up of your scale chyma cells. So the scale chyma cell, it provides support to it, right? So this is how the dicot stem looks like. So next, let us learn about the theory what is given in your book. So the transverse section of a typical young dicotyledonous stem shows the epidermis, which is the outermost. Yes, we know that epidermis is the outermost layer or the protective layer of the stem, right? And it is covered with a thin layer of cuticle. It may be bearing trichomes, as I have shown that this epidermis, after that there are these layers, isn't it? These are nothing but your root hairs or your trichomes, right? These are your trichomes or your root hairs. They may be bearing trichomes or they may not be bearing the root hairs. So the cells arranged in multiple layers between epidermis and pericycle constitute the cortex. So as you have seen that there are three types of cells, and the arrangement of these cells, what we call them, that is your cortex. This arrangement is nothing. These three cells we are calling it as cortex, isn't it? Yes. So next let us see. It consists of three subzones. As I have said that the cortex is having three layers, that is your hypodermis consisting of a few parenchymatous cells just below the epidermis, and which provides the mechanical strength, and the cortical layers below the hypodermis consisting of a parenchymatous cells. Okay, on top of the hypodermis, that is your epidermis, isn't it? Which is bearing the parenchyma cells, epidermis. Then on below the hypodermis, isn't it? So that is below the epidermis is your hypodermis, which is bearing the thin walled cells. Always the parenchyma cells are thin, isn't it? So they are having thin walled cells, that is your parenchyma cells, with conspicuous means which we can see, that is visible. and intercellular space, and they have got intercellular space also in between them. So the innermost layer of the cortex is called the endodermis. So as you have seen that the innermost layer, everything inside it we call is endodermis, right? And this endodermis, whatever everything is present inside the endodermis, what we call it? We call it as stem. Isn't it? Everything inside the endodermis we call it as stem. Yes. So next is the cells of the endodermis are rich in starch grains. Okay. So the cells are rich in starch grains. They are responsible for storing the starch grains, and the layer is referred to as starch sheet, which is absent in case of your monocot stem. This is absent. You will not find this in case of your monocot stem. So from here you can differentiate also difference between your monocot stem and dicot stem. What we see in case of dicot stem, we are looking that they are made up of your what? Pericycle is made up of your sclerenchyma cells, isn't it? Whereas in case of your monocot stem, it is made up of your the parenchyma cells, and in the monocot stem, in the dicot stem, we can find the starch sheet. Whereas in case of dicot stem, the starch sheet is absent. So pericycle is present on the inner side of the endodermis, and above the phloem is the form of semilunar patches of sclerenchyma. So as we have seen that pericycle is present on the inner side. So look here, where is the pericycle? This pericycle is always in the inner side, right? And above, that is pericycle, it is made up of your sclerenchyma cells. And in between, we can find in between the xylem and phloem, the medullary rays too. So this is all about your dicot stem. So yeah, in between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute the medullary rays, which I have shown that in between the vascular bundle. So if this is a vascular bundle, so near the pericycle, we have seen the vascular bundle, right? So here you will see some rays near the xylem and phloem. So this is your xylem and phloem, right? This is your xylem, phloem. Here, these rays are called as your medullary rays. These rays are called as your medullary rays, right? A large number of vascular bundles are arranged in a ring, and ring arrangement of the vascular array bundles is a characteristic of your dicot stem. So this ring arrangement, how it is arranged, it is the characteristic of your dicot stem. So each vascular bundle is conjoined open. That means they are having what? They are having the cambium. Here you will see the presence of your cambium. All right. So this is open. That is cambium. We will find it. Cambium is present here, isn't it? So here the xylem and phloem. Xylem is always present inside. Phloem is present outside. So see xylem and phloem, and we will find the cambium here. All right. So when we find the cambium, we call it as conjoined open. Conjoined open means cambium is present with underged protoxylem. So here we see the underged protoxylem. What is this underged protoxylem? Means what? Means if this is a vascular bundle, here what happens? Here let me show you that the pericycle will be that is protoxylem will be inside. This is your protoxylem. This is PX I am writing. This is your protoxylem, and this is your metaxylem. Metaxylem will be towards the periphery. Okay, metaxylem will be towards the periphery, and protoxylem will be in the center. We call this condition, which we find in case of stem, we call this condition as endarch, not exarch. Endarch means what? Endarch means your protoxylem is present inside. Is that clear? Protoxylem is inside and metaxylem is towards the periphery. So we find it in case of your stem, we call it as endodermis. A large number of rounded parenchymatous cells with large intercellular space which occupy the central portion of the stem constitute the pith. Okay, so that is your pith. So as you can see, this is the picture which shows the epidermis and this is your pith, isn't it? This is your pith and this is the hypodermal layer. So this is the diagram which I have drawn. This is same. Now let us see the monocot stem. It is also same. So in case of your monocot stem, what happens? They have a circular shaped hollow axial part of the plant which gives rise to nodes, internodes, leaves and all the flowers and the basal end. So in case of your monocot stem, how you will what you will find? So see here, you will see many vascular bundles. So you will see here many vascular bundles. So vascular bundle will be many or more. So here you will find only one layer. You will find only one layer, that is your hypodermis. Yeah, and this outer layer is your epidermis and this is your hypodermis. So here you will not find the pith. Can you find the pith here? Pith is present? No. All these are vascular bundle will be present. So these are your vascular bundles. Okay, that is your xylem and phloem. And this is your ground tissue. That means your parenchyma, sclerenchyma and parenchyma. All these are filled with your ground tissue. They are filled with what? They are filled with your ground tissue. Here we are not finding the pith. So what you can write? Pith is absent. Pith absent. Then we cannot find the starch sheet, isn't it? In the dicot stem, we have seen the starch sheet, isn't it? So here we cannot see the medullary rays also. Medullary rays also absent. Pith also absent. The that monocot stem. So see, the monocot stem has a sclerenchymatous hypodermis. So in them, the hypodermis is made up of your sclerenchymatous cells. Whereas in case of your dicot stem, they are made up of your parenchymatous cells. And they have large number of your scattered vascular bundles surrounded by your sclerenchymatous bundle sheet and a large conspicuous parenchymatous ground tissue. So when we look here, so you will see so many vascular bundles. All these are nothing but your vascular bundles which are made up of what? Which are made up of your, that is which will be having large conspicuous parenchymatous ground tissue. As I have shown here, these are having here surrounded by your parenchymatous ground tissue. This is your epidermis which we find in all parts of the plant. So this is all about your monocot stem. So in this session we have learned about the anatomy of your dicotyledonous stem and the monocotyledonous stem, right? And also we have covered the vascular tissue system. I hope you have understood each and every concept what is discussed in this present session. If you find this video useful, do share it with others and also don't forget to like and visit to our website where we will be finding the PDF notes of all these topics. Visit to our website, that is www.v-learning.in, where you will be getting the PDF notes and all the pre-recorded videos too. So this is all about today. Be connected with Vista's learning. I will meet you in my next class. So keep learning and be safe.